Millions of Americans suffer from peripheral artery disease and coronary artery disease, which are caused by a buildup of plaque and can lead to insufficient blood flow. These diseases may be treated with a drug-eluting balloon and a percutaneous transluminal angioplasty. Our design allows for in vitro evaluation of drug-eluting balloons and performance of percutaneous transluminal angioplasty in relevant physiological conditions, along with evaluation for cellular response to treatment. Current perfusion bioreactors are expensive to fabricate, are not automated, and do not closely replicate physiological conditions. Thus, the objective of our project was to develop a cost-effective, automated tubular perfusion bioreactor to facilitate endothelialization on a surface-modified silicone tube and to model in vitro percutaneous transluminal angioplasty and test a drug-eluting balloon. The solution we have proposed is to use a bioreactor to grow a monolayer of cells in the lumen of a polymer tube, which resembles the physiologic conditions of a blood vessel. To execute our solution, we developed an automated perfusion bioreactor that can be operated 24/7, is connected to the Internet of Things to allow for remote feedback and operation for maintenance of physiologic conditions, and the system also features a user-friendly design that is customizable to different applications.
The automated bioreactor we developed has the flow created through a peristaltic pump, and then the media is delivered through the sensor reservoir that measures the temperature, pH, and flow of the system. After it passes through the reservoir, the media flows through two coated silicone tubes and back into the media reservoir. The silicone tubes are coated with polydopamine as well as collagen type 1 and 3. After each test, the system has ethanol flown through as well as a UVC light turned on to sterilize the system and prepare for more tests. The testing process for our bioreactor began with sterilization of the system, and then the media was perfused through the system and the cells were inoculated. The system was run for 12 hours before we removed the control tube and validated the presence of endothelial cells in the lumen. Fluorescent staining and light microscopy analysis were completed to determine cell location and morphology, and the cell count and viability were obtained by dyeing the cells with trypan blue to sample on an automated cell counter.
And so our final product can be seen at the top of our experimental results. Currently in all three pictures, the acrylic sliding doors have been removed for visibility. From the front view of the system, you can clearly see that our test site that holds our experimental tube is at the top, and the control tube is right below that, which is used to verify a monolayer of cells before performing the PTA balloon testing. Right above that is our one-time use cell inoculation site. And so after 12 hours of perfusing the cells to the system and staining them with red cell tracker, our results can be seen in the figure labeled coated silicone tubing. This image was taken with the cells still intact on the surface of the modified tube. The positive control reflects the results seen after growing the same cell line inside the CO2 incubator, but with optimal conditions and no flow. The viability of the positive control was 99% and 93% for the experimental group using the silicone tubing. The control group's proliferation after 12 hours increased by approximately 16.97% and a 7.61% increase for the experimental group. We attribute the lower proliferation of the cells growing in the modified silicone tube to the pressure experienced on the inner silicone lumen as well as the constant flow of the cells throughout the system. We also observed an uneven layer of cells growing on the silicone tube. Instead, most cells grew at the bottom of the silicone tube. This can be caused by the horizontal orientation of the test site and the gravitational forces acting on the cells. In conclusion, we successfully designed and fabricated an automated tubular perfusion bioreactor that's capable of growing cells under physiological conditions, all under $600. We believe that this system has the potential to aid researchers in predicting the human cellular and intravascular responses to drug-eluting balloons before animal and clinical trials. However, the next generation of tubular perfusion bioreactors will strive to replicate and integrate human anatomical geometry of the arteries that are typically curved, bent, and angled. Upgrading our in vitro design to an ex vivo design could also prove to be useful in closely replicating in vivo conditions. Lastly, once a monolayer of cells has been confirmed on the surface of the modified tube, the next step would be to test drug-coated angioplasty balloons with the bioreactor. We would like to give a special thank you to Dr. Nguyen for helping us throughout this project and providing all the necessary materials needed, as well as Mr. Luis Soto Garcia for his mentorship and involvement in this project. Thank you.